Earlier, we talked about getting data from the heat map view using our cursor to go over a specific region, which gave us a table of genes that we could see. Now we're going to talk about what drilling down into a gene of interest. So let's click on this one, which is this, just because it's named. There's no particular reason to do this, but why this gene and not the others? I wanted something in the middle, which is this particular gene ID 358. That's pretty much in the middle of this group. When I click on that, it populates the vertical green bar with possible downstream actions. This time, we're going to click on Feature. That's going to open a new tab that shows all the information we have about this particular gene. In PATRIC, the unique identifier is this, which we've talked about. This is the unique identifier for the genome, which is this Brucella microti genome. This is a breadcrumb across the top that tells us exactly where we are. In RefSeq, this genome, this particular gene is known as this. This is the gene symbol. This is what it's called in PATRIC, and it's a coding sequence gene. This is the protein ID, and if I were to click on this and say open it in a new tab, it should take me to that gene in RefSeq, and they've updated it recently, so that you could follow the links there. This is the gene ID, this is the protein family, this is the global family, and here you have more information about the genome itself, more information about the exact location of this gene, and you could see the sequence, how big the gene is, and the protein and the length. Also here it shows you where the gene is in bright red and who the neighbors are around it that you can see by scrolling over. Also, you can go to different database repositories and look for information about this gene. One of my favorites is the CDD, sorry. Everybody's got CDC on the brain now with the outbreak. But let's click on Conserved Domain Database. This might take a while to upload. Oh, there we go. And that's showing what the information is about this gene. I really like this this particular resource. It's really good. You could also look at string to see if there are any known protein-protein interactions for it. None are found. Not surprising because it's kind of obscure little gene. But pay attention to these particular links to external tools in PATRIC. I've also talked to you recently about how you can add a genome to a group. You could add this to a feature group. If I wanted to see how many of the PATRIC Brucella genomes had this particular local family, I could click here. It would open a new tab. We would wait a few minutes, hopefully not too long, for this to load. And it tells me that 155 of the PATRIC Brucella genomes have that particular protein family. You could do the global family, which goes beyond the genus boundary. So let's click on that. It should be bigger, but let's see how big it is. Oh yeah, 1,720. So this gives you an idea of how many different genera have this particular protein in them. You could see how many genomes have the exact same sequence. I'm going to click on this and open a new tab of this, and that's 99. So this was the protein families, the local protein families, which was 155. 99 of those have the exact same sequence. I'm going to close some of these up so that we don't, so we'll close some of the tabs. So I really like the feature page for this. We also have different tabs, the Genome Browser and the Compare Region Viewer. What I want to talk to you today is about the Compare Region Viewer. Usually when you're doing bioinformatic analyses, it all centers. You have to go to extra lengths to get beyond the gene. It's easy. You can do the genome comparison, you can do the gene comparison, but doing the gene neighborhood can be a little bit difficult. We have this function, which is the Compare Region Viewer, that shows that. So let's click on that. We have to wait a few minutes for it to open. And what this is showing us is that the gene that we came in on, which is this INC-P type DNA relaxase TRA1 gene, it's here in red. And you may ask, why are there two different bars? Well, every gene, when it's starting to draw in the gene neighborhood, when you launch this tool, it wants to see how strong the blast hit is to that particular gene. So because we started with microDE, it's a perfect hit, right? And it's a really strong, this represents the blast hit, which you can see as you mouse over it. And this represents the gene, telling you the location. As we grow down the Brucella and suddenly we get to this one, the blast hit is less strong, and so it's lighter. And look here at this one. It only had a partial blast hit to that gene. Okay, so what else are we seeing here in the Compare Region View, and what is this doing? It's taking the upstream region and the downstream region that equals 10,000 base pairs. It takes that entire region and it looks across, in this instance, the reference and representative genomes in PATRIC to see how tightly conserved, to find the closest matches, not for the gene, but for the neighborhood. So it's looking for this entire neighborhood. And the reference and representative genomes are something that GenBank has and assigns. They're the gold standard or high quality genome, and they're showing the ones that match that in Brucella. And you can see in the upstream regions, the colors are all similar. These genes, if I mouse over them, they're hitting the same gene. And these are all the same, annotated the same way. Some of them, like this one, this little hypothetical gene, is missing in the neotoma, but it's present in most of the others. And then you can see in the downstream region, these are all hypothetical genes, so it's coloring them all the same color. When you get out of Brucella, you notice that while this gene stays the same, that's as far as it could get. The rest of the neighborhood is totally shot, looking at the reference and representative genes. Let's look at this a bit more closely. We have Brucella microDE, which is the genome we were looking at, this NVSL strain, neotoma, canis, suis, seti. Well, I know that there are different Brucella reference and representative genomes. Let's go back to the heat map view that we launched a while earlier, and remember when we marched through this, the abortus, there are reference and representative abortus genomes. Those appear to be missing, and the melitensis genomes are also missing. Now, when we first launched this tool, we were just using a subset of all the genomes of Brucella that are available in PATRIC. So let's see if this holds if we extend it. So one of the things we want to do is go from reference and representative to all public genomes. And notice we're looking at cross-genome families. That would be important here because it's showing how strongly this is conserved across the genus boundaries. Remember that this section of Brucella, let me go back to the heat map view, this is a known genomic island. So we're trying to see which other genomes in PATRIC might have features of this island. So we'd want to keep it across genome families. You could change the size if you wanted to, and then click Update. Now that always takes a long time, so I launched it earlier. I cheated to show you that. So these are across all the Brucella genomes, and I wanted to see how many abortus genomes had this. So there's 1, 2, 3, only 3 of these genomes, which we'll explore later in a different part of the analyses. So it's sort of confirming that a lot of these genomes are that this is unique to outside of abortus. And I just want to, scroll all the way down to the bottom. It's a very long page. We get to Brucella suis, and then we get to Serratia. And look here, we're seeing on this 5' end of this region, we're seeing a lot of similarity here. Once we got out of the reference and representative genomes, we can see that this is conserved. We can see this 5' region that goes all the way up to the protein we came in on, but after that, it falls apart. So it looks like for this particular genomic island in Brucella, looks like these guys are unique. Remember when we were looking at the reference and representative genomes, they were missing this in the 5' region too. But these particular genomes have a piece of it. So I think that there's something going on with horizontal transfer, and this part, this 5' end, is pretty conserved in across these genomes, which aren't a lot. You know, PATRIC has over 300,000 genomes, so now we're talking about 5 or 6, but they seem to be having the same genes in this area, but once you get here, things change up quite a bit. So it's an interesting way to look at things. In the next instructional video, we're going to talk about using the Genome Browser and capturing data, the sequence that corresponds to this area, and trying to demonstrate how you can determine, how you can verify that something is really missing in a different genome. Thanks for listening, and thank you for using PATRIC.